Bharadwaj, and you all are watching Medically Speaking. So today we are going to discuss about RNA platform as well as new variants. So we have with us Dr. Rakesh MIshra, who is a director of CSIR CCMB. Sir, welcome to Medically Speaking.
Thank you.
Sir, we all are talking about, a good behavior, especially maintaining a social distance, wearing a mask. But still, do you think the way, mutation is taking place, it is a cause of concern?
So the mutation is a natural process. It will take place and it doesn't really depend on behavior. What behavior does, though, in this process is that we prevent the spread of these mutations. And that is where if we are following certain discipline, we can limit the transmission of the virus, because virus goes from person to person. There is no other means. So only when two people are together and they are not following discipline, the virus will infect. So during this process, when virus is multiplying in an individual, it naturally creates mutations, and some of them may be troublesome, and that's why we should not give much chance for the virus to mutate and spread and create more problems for us.
So do you think this South African variant is more dangerous?
See, at the moment, there are no, it's not confirmed whether the virus has more mortality. Some of them are supposed to be more infectious, which might be true, but may not necessarily mean more problems, except that it may create a crowd at the hospitals, and more number of people may get infected at a given time, and therefore more playing ground for the virus to come up with new variants. Some of the new variants are now suspected to be more resistant to the vaccines, in the sense that they need more antibody for the inactivation in the experimental conditions in vitro neutralization assays, which is something to worry about. At the moment, though, none of them have been confirmed that they will not respond to the vaccines, but it can happen in the future. And that's the biggest problem of these viruses, that they spread fast and they create new versions of them and come up with the new problems. So it's now we are noticing few, but in fact mutations are happening constantly. There are several thousand mutations they are documented, but since they are not of much consequence, they are not noticed. Some of them, when they start to become more infectious, then they get more attention. Other important thing in new mutants can do is that sometimes our tests may not work in some of them, because tests are, particularly RT-PCR tests, which are depending on the sequence of the virus. And if at the correct place where the primers are binding to detect the virus, if that gets muted, we might miss them out. And so therefore mutations are something to be very watchful, and that's why extensive sequencing and genome survey is very important to be prepared and have an idea what is happening in the population.
So especially if we talk about this South African, this mutant variant, so are we screening for this particular variant in India?
Yes. So now the India has a policy to sequence 5% of all new cases, and sequence all the positive that have travel history, they come from abroad. So and the sequencing is neutral to any variant. It will detect any variant, old, new, South African, UK, or even the variant which may be very new, not known before. So sequencing is basically reading the genome of the virus. So, and the government has put a plan to sequence 5% of the new variant, which is a decent number to get an idea if there are new things coming.
Sir, yes. And sir, do we have those test technology, especially to sequence such viruses?
Yeah. Our sequencing technology and expertise and to analyze these sequences is far beyond the challenge that virus genome poses. It's very small genome, and you can sequence hundreds or thousands in a go and analyze them in very short time, in less than a day. So those things exist. The key thing of genome sequencing of the virus is to get the sample from in a very well distributed place, so we cover the whole country. If you sequence thousands of genomes from one mohalla, one gully, it doesn't mean much. They are likely to be the same. But you should be able to sequence samples from all parts of the country, all districts, all blocks, all cities and towns, and so on. So whatever positive comes, a certain percentage of those. So we will know if at some place a new variant is emerging and is spreading, because we should sequence at least once in a week these samples with certain staggered plan. So we will know if there is a new variant which is coming and spreading in that region. So then we can immediately put more restriction in that region and block it from spreading. And that way, spread of a new variant, bad or worse, can be prevented. So that's what is the whole idea of surveillance. So sequencing capacity is not an issue. The issue is strategy and getting samples from the field. Imagine if you want to sequence samples from the entire country as widespread as you can, so that even at very early stage, before virus becomes a very large number of people is detected, is quite a challenge. So that is an issue which I think there are plans being put in place to take care of that.
Yes, sir. The way mutation is taking place, and the lot of scientists are talking about that these particular variants, especially we talk about UK strain and the South African strain, are becoming dominant.
Yes, so that actually happens. I mean, we know these are the reason they become dominant is because they spread more, and therefore you start seeing them more. That doesn't mean that they are more dangerous. For instance, at one time in India, we had identified Southeast Asian origin variant of this. We used to call it A3I, which was a very high percentage, more than 40% in a certain part of the country, including south of India and Delhi. But we knew that that's although it's a new variant, a new clade, but it has some deficiency in replication, and it might not be able to compete. So it disappeared gradually. Now we don't find A3I. Similarly, now there are new clade emerging, which we call for E440K. It doesn't matter, it's just that it's a new variant, and that is spreading most. Now close to 50% of the people, of the positive in south of India, most probably in other parts of the country, are this variant or clade or mutant, if you want to call. But looks like this is also not more dangerous in the sense more mortality or more symptomatic and so on. It's just that it has certain advantage over the other existing variants, and therefore it is seen in more number of people and gradually expanding. So this keeps happening. The problem will come when suddenly one of these variants is also ignoring the vaccines or the antibodies. Then we will have difficulties, because then the vaccines will not work against this, like happens in common flu, that each year you need a new vaccine. But if you need a new vaccine every 6 months or 1 year for COVID-19, that will be just untenable. So that is why the idea is to monitor very closely, prevent the new variant from spreading, and create as many roadblocks as one can by mask and social physical distancing and hygiene practice, so that we can slow down the spread of the virus. So we are now actually in a position which we can call it a winning position. The number is falling, hospitals are not crowded, and recovery rate is very high and so on. But at this time, and the vaccines have come, they are now being implemented, and we understand this disease more. We know exactly how it spreads, how much it can spread through air and those sorts of things. So this is time we should go for a kill and attack the disease from all aspects: social aspect, masking, distancing, practice a certain discipline, using as much vaccine as we can, convince people that it is good for you, it is safe for you, and close surveillance by genome sequencing and watching out for the new emergence of new variants. If we do all these things together, in a few months after time, we can certainly control the disease well. But if we don't do this, we depend on, oh, vaccine has come, so now there's no problem. But we know the vaccine can be effective. Maybe 70-80% of the people, 20-30% will not respond to vaccine. Even the best vaccine, 5% will not respond. There are also 5% is people. And new variants come, they may be less effective. Vaccines may be less effective or not at all effective against new variants. So these are all tools, important tools like vaccine and all, but they should be used, but we should not drop our guards because they have come. So all these things will have contribution, and we succeed only if we work in a concerted manner and follow, use all our ammunition that we have against the virus. The virus is in a weaker position now, numbers are falling, and that's time to kill it. If we let the virus recover, let the virus spread more, it will have more chance of coming with more troublesome variants. So it's very, very important to be watchful and really, as I said, go for the kill, using all possible ways of controlling it.
Go for the kill is very important. Sir, one very good news which I heard about it, that is a CCMB is working towards putting together a RNA platform which will prove beneficial for the development of indigenous RNA-based vaccines. So what is an RNA platform? What exactly it is, sir? And how it is going to be helpful?
Yeah, so see, RNA was supposed to or expected to have therapeutic value many decades ago, and people have been trying to use RNA to control genes or use them as a drug because they code for proteins, and or they can go and inactivate a gene that you desire to be inactivated. But for a variety of reasons, we'll not go into details. This technology was not taking off. It was causing non-specific effects. But in several decades, people have been using actually mRNA vaccines against cancer, and lots of development has taken place. People have modified the RNA in such a way that it is not immunogenic by itself, and therefore there is a specificity in its action. And you can pack viruses. RNA are generally very unstable. So now people have found ways of making them more stable and then packaging them in lipid nanoparticles. All these recent technologies have made RNA therapeutics as a very attractive possibility, although they're not drugs that are already in market for large scale at the moment. What happened during the COVID is that mRNA-based vaccines were pushed and they showed very strong result. And considering the emergency, considering the situation, they have been given approval and for emergency usage, and that was a big boost for RNA therapeutics, I will say. And these vaccines are doing well. The results in third phase have shown good results, and now they are already being given to people in Europe and America. So this has been a game changer from the therapeutics point of view, because this means that if today I want to make a vaccine against a different variant of this virus, in 3-4 weeks I can have a new version. Is that fast? And you don't need setup, you don't need many lots of safety labs for this, because you don't grow any virus, you don't grow any protein, you just design a construct. So there are certain modules for RNA therapeutics. So for example, you conceptualize and design your target and do what we call as molecular cloning, to make a DNA construct of this. Regularly all the labs do it, molecular biology labs do it as a routine, and then make this DNA molecule called plasmid and grow in a large quantity in bacterial cells, which is easy. In a couple of days, you can go very large in grams of this. And then this template of DNA can be used to make RNA using the process called transcription. And this can be done in test tubes, so you don't have to grow in an animal or virus or anything. And whatever you have designed or conceived as a target, as a step one, can now be delivered as a mRNA or RNA for that target. And then this RNA can be packaged in a lipid nanoparticles and ready to go for injection in people. Of course, clinical trials and tests and those things need to be done. So to come with the new versions of any therapeutic options of an existing vaccine or drug becomes very simple, and a small setup can make really large quantity of this, so it can be tested very easily. So this enables us to come up with the vaccines or other therapeutics in a very fast and very precise manner. CCMB-like laboratories have most of these technologies already in place. What we are doing is setting up a RNA therapeutics and vaccine platform in which all the modules of design, making construct, growing the plasmid, doing transcription to make the mRNA, stabilizing the mRNA by chemical modifications, and packaging it in the nanoparticles being put in one platform, so that from concept to delivery of a molecule that can be tested, the formulation that get tested can be made in on one platform. So this will not only be for the COVID vaccine. This can be used for rare genetic diseases where you need to provide certain protein which is defective in the disease. And India has 70 million people affected with rare genetic diseases, for which, for many of them, there's not even a name. Forget about the cure. And so these are things which might go a long way in addressing those because they are India specific, and because the numbers are not in millions and millions, so their market will be smaller, and bigger companies will not come into the into picture. So, but to help these people, these kinds of therapeutics approaches will be very useful. So that's why, with a long-term plan, CCMB is setting up this platform. Lots of CSIR labs are helping in making the modules in different modules more efficient and indigenize them completely, so that we should be able to endogenously prepare, conceptualize and prepare a drug or a vaccine at very short time using our technology. That's the purpose of this the platform that CCMB has just begun implementing.
So maybe it will take a long time, sir. So, but we can say that, you know, the rare diseases treatment, which was not available in India itself. So we can expect, you know, it is a kind of positive sign towards that.
Right, yeah. So rare diseases are an issue which we are very serious about, and therapeutic is one approach for them. When the child is born, then nothing much can be done, and you have to provide the therapeutics. But we are also working very hard to develop markers for them and design cheap assays for a few hundred rupees, so that parents can be tested and given advice if they need to be careful about the child, and whether they say what is the possibility of a child having the rare disease, because once you set up the markers, you can figure out, and then you can counsel them appropriately, or they should do prenatal diagnosis and take a decision depending upon the genetic status of the embryo. So there are multiple ways. The therapeutic is the one that we are talking currently, and RNA therapeutics is a very good option for that. It gives the flexibility, it gives the options, and multiple trials can be done at short time, and so on. So it's something which I think is a technology for the future, and it may go a long way.
Thank you so much, sir, for talking to Medically Speaking and sharing so much important information with us. Thank you so much, sir.
Thank you. Pleasure talking to you. Thank you.